So remember that you have this membrane, and it's like this, and the individual lipids can sort of diffuse in the membrane. They can't flip-flop from one side to the other, but they can easily sort of switch places with their neighbors in the top layer of the bilayer or the bottom of the layer. So that the ease with which they can move relative to each other controls the fluidity of the membrane. And that's important because in order for something to move through the bilayer, it's got to push these various lipids out of the way and move through. And so the more fluid they are, the more easily they can move relative to each other, so the easier it is for something to go through. So the less fluid a membrane is, the less able you're going to have small molecules diffusing through. So increased fluidity, more diffusion through, decreased fluidity, less diffusion. And so cholesterol, basically when cholesterol gets inserted into these lipid bilayers, it tends to cause the tails of many of these molecules to straighten out. And that does a couple of things. By straightening out the tails, you can see here in this little cartoon, they're straighter than this. It causes the membrane to get a little bit thicker. But more importantly, by making the tails sort of compact, these guys can now pack more closely together. And by packing more closely, they interact more strongly. So the more packed they are, the less fluid they are. Okay? So a big thing that the cholesterol does by causing this change and by also interacting with more than one of them at a time is to really decrease the fluidity of the membrane. Okay? And so that can have effects it can affect how things go through that membrane. And in particular, we wrote a paper studying the effect of altering cholesterol content on mitochondrial membranes. So the finding was that there's this protein called caveolin, and caveolin is a protein that jumps around inside of cells from one membrane to another, and it can carry cholesterol with it. And so if you have a mutation in cholesterol, then a mutation in caveolin, then the fluxes of cholesterol are altered in the cell, and that can lead to different content of cholesterol in some of the internal membranes, in particular like the mitochondria, and that alters their function. And that can lead to all sorts of diseases. So if you go to my website, if you have any interest, if you go to the website, you can get the paper and read it or not. Okay. So that's cholesterol. So I've talked about the lateral diffusion allowing the fluidity of the membrane. And I had mentioned, I think, last time a little bit, that there is asymmetry in terms of which lipids are on which side of the bilayer. So typically, phosphatidylcholine and sphingomyelin, PC and PM, are on the extracellular portion of the plasma membrane, and PE and PS are in the inner one. Okay? And so that was created by how they're stuck in, but also this flipase that controls how much they're flipped from one to the other. And that's important because, for instance, PE, or maybe it's PS, let's see. PS can be used as a signal. So usually PE and PS are inside, but when the cell undergoes apoptosis, it commits suicide because something bad is going on inside of the cell, and it does that typically to protect all the other cells. So for instance, it's infected with a virus and it wants to kill itself before the virus spreads and goes to other cells. So one of the things that is triggered in the apoptotic pathway is to flip to have a flipase that's activated that flips PS from the inner membrane or from the inner surface to the outer surface. And that flip and having PS on the outer surface then signals to macrophages, which are cells that come around and eat things, to eat the cell. Okay, so the PS flip is a signaling process that says this guy's bad, kill it off. And so that's one example of a signaling event related to lipids. You can also have more local signaling events where if you accumulate a bunch of lipids together of a particular sort of phospholipid, that can provide a small region in the membrane where you can specifically recruit a protein or proteins and sort of start a local signaling event. And so, for instance, if you have a bunch of PS together, then that can recruit ankyrin and other things can recruit other proteins. So you can have sort of localized signaling events on membranes by which membranes you have where. and sort of exactly how you get a bunch of, so typically these bilayers are mixed lipids, and so how you accumulate a particular phospholipid in a particular place is not so clear. Okay, and that's an area of active research. One thing that can certainly happen is that certain lipids like to have certain curvature. So for instance, phosphatidylethanolamine, its tails tend to spread out a little bit. So if you put a bunch of them together as part of a bilayer, that portion of the bilayer, if you don't have PE on the other side if you say put PC on the other side, then you can get curvature on one side and not on the other. And so that basically can be where you have a curve or curvature in the membrane. So not only can localizing PE to one portion of the bilayer cause curvature, but alternatively, if you physically cause curvature, you're going to have PE migrating into that region because that's going to be energetically favorable. Okay? But once it migrates into that region, it's going to take less energy, less force to maintain the curvature there. Because you're initially having to do a lot of work to keep it curved because it wants to pop back because it doesn't like to have this curvature. But once the PE goes in, then it takes less. So you can have things go both ways. One thing is if you deform it, then you have diffusion in, but also if you sort of locally somehow get PE to stay there, then you'll get curvature. So they feed off each other. So that brings me to a Clipper question. And this is a subtle question. So I encourage you to read it carefully and think about it before you answer. And we'll have 2 minutes. So I will tell you that the majority of the first class got it wrong. But I think it's good to think about it. So I'm going to stop at 2 minutes, so you've still got time. Maybe I'll start at 155. I don't see numbers going up. Okay. Well, so you guys did better than the last class. The last class, everyone thought B, and this is sort of more of a mix. Correct answer is D. So let's discuss why. Okay, so why is this? So there are really 3 sets of things. So A and B are mirror images of each other. So A basically says you're going to look at fluidity of the membrane and you would see increased mobility. That's not right because you're expecting that if cholesterol is going to be there, it's going to decrease mobility. So that's wrong. B is right in the sense that if nothing else were to change, you would expect that if you put additional cholesterol into the membrane, you would have decreased fluidity. decreased mobility. And so that answer is in principle right. So why don't I think it's the best answer? Well, the reason that you don't use C is that when you grind things up, basically you're going to get membranes, you're going to get internal membranes, and you're going to get plasma membrane, and so you don't know what's what. And so just if there were increased cholesterol in the membrane fraction, you don't know which membrane it is, and this specifically asks about the plasma membrane. So C isn't specific enough. So now D basically looks at, actually measures the amount of cholesterol in the membrane using a cholesterol binding dye. Okay. And measuring the average intensity. Okay, so now this directly measures cholesterol, and this measures a side effect of the cholesterol. So why do I think this is better than this? Okay, the answer is that D is not a definitive experiment. Okay? It is true that if the cells incorporated cholesterol and did nothing else, and did not respond in any way, the mobility would be decreased. So that's right. But you can imagine 3 other scenarios, I think, where you would increase, where you would have the wrong conclusion. So scenario 1 would be, suppose that the cholesterol was detected by the cell, and the cell altered, in fact, used the cholesterol in internal membranes and not in the plasma membrane, but caused some adaptive response, so it changed other lipids in the plasma membrane. So changing other lipids in the plasma membrane could also alter the amount of fluidity, having nothing to do with cholesterol being incorporated into the plasma membrane. So you could have had an adaptive response of the cell, which changed the cell's fluidity, which didn't directly reflect incorporation of cholesterol. So that's sort of one way that you would have gotten, you could have gotten the experimental result that you expected, and you would have had the wrong conclusion. Okay? A second thing that could have happened is it could have been that the cell was monitoring the membrane, and so when you did in fact increase cholesterol in the membrane, but then you decreased something else to keep the membrane fluidity the same. And if that was the case, then what you would measure is that there was no change in fluidity, and so you would conclude that there wasn't any incorporation of cholesterol, but in fact there was. It was just that there was a compensation. Okay, so both of those would give you the wrong answer here, even though it was a reasonable experiment. But here, you're not looking at the downstream effects, you're just directly measuring cholesterol, and so this is a much more direct answer to this question. Yes?
But couldn't, since you're not looking, since it's not a quantitative experiment, can't you just also expose and then release the heat, and then test for that as well? Would that be a lot easier?
But you don't know, but any adaptation that happens in the cell could have affected membrane fluidity and could give you what you observe without it being due to cholesterol. Right? So, because the different membranes, I mean the different lipids contribute differently to membrane fluidity, and so you could have had adaptation in the cell due to the presence of cholesterol that has nothing to do with direct incorporation of cholesterol into the plasma membrane.
I always remember cholesterol, needs some heat resistance, and so I never read about any other lipid that does that as far as
So you could do that, but again, it's less direct. So if you had a dye, as is suggested here, if you have a dye that directly measures the amount of cholesterol, that's a much better experiment than trying to look at the second thing. Okay, yes?
What's the point of the second sentence, Josie?
Okay, because, so if you don't change the medium, you're going to get a lot of background. So if you use a fluorescent dye that is going to bind to cholesterol, if you don't change the medium, when you put in this fluorescent dye, it's going to bind to all the cholesterol that's floating around in the medium, and you're just going to get a lot of fluorescence too, and you're going to be able to see nothing. So that's why it's there. Okay. Oh, yes, sorry.
Wait, so what, if you increase the temperature, what exactly, what effect does it have on cholesterol?
So, increasing the temperature, let's see, so cholesterol does a couple of things. It it does give you, I guess, temperature stability, right? So if the cell isn't doing anything else, if you saw stability and not so many changes as a function of cholesterol, I mean, as a function of temperature, then that would suggest that cholesterol was there. It wouldn't necessarily, but then you're trying to look at changes.
That's like fluid-wise, like does it make the membrane more fluid?
Cholesterol makes the membranes different. It decreases fluidity. And so if the only thing that had happened was that you incorporated cholesterol, nothing else happened, B would give you the right answer. It's just that you can imagine other scenarios which could also happen in a live cell where you would get, where doing this experiment only would give you the wrong answer. So that was why I wanted you to think about the different alternatives. But this was tricky. And okay, so that brings us to things going through the cell. And the key point is that to get things into the membrane or through the membrane that are large polar molecules or ions, you need active transport, and we're going to talk about how that happens. But basically the idea is that in order for things to get transported through, you need to create an interior space in the membrane which doesn't look hydrophobic. Okay? And the way you do that is you have proteins that are integral membrane proteins that cross the membrane and create some sort of interior space so that molecules can go sort of through the proteins and not be exposed to the lipids. Okay, so we're going to discuss what are the properties of a protein that's going to get embedded in the membrane. Okay, so those things that get embedded in the membrane and cross it all the way through are called integral membrane proteins. That's different from, say, lipid anchor proteins, which say stick a prenyl group in and stick partly through but don't go all the way through. Okay, and that's different from peripheral proteins that say just bind to other membrane-bound proteins. So the integral proteins, what they've got is they've got a hydrophilic portion over here and a hydrophilic portion over here and a hydrophobic portion in the middle. Okay. And what that means is that the energy cost to remove them from the membrane is huge, because the hydrophobic region in here doesn't want to go over here, so there's an energy cost for removing the hydrophobic region from the lipids. But in addition, there's this hydrophilic domain over here, and there's a big energy cost if you try and move the protein up of putting this hydrophilic domain in the lipids, in the hydrophobic. So there are two energy costs, right? So the fact that you've got a hydrophilic domain on each side means that you can't pull it through. Whereas this guy, you can pull it straight out and there's much less energy cost. Okay, so that's why you have these integral membrane proteins, and they can, if you have them crossing the membrane, you can get some sort of internal space, and they're going to be the ones who are transporters. So the first question is, well, what are the properties of a membrane protein you need? And the key thing is, it's got to be hydrophobic, because it has to be happy in the membrane. And typically, one of the things that is used to span membranes is an alpha helix, which has 20 to 25 amino acids. And these 20 to 25 amino acids have to be hydrophobic. And so that basically ends up being about 3.6 nanometers, I think, something like that, all in length. And so what are the hydrophobic amino acids? Well, here they are. So phenylalanine, isoleucine, leucine, these are pretty hydrophobic. The positive guys here are the hydrophobic ones. Alanine is a little bit hydrophobic, and then there are other guys. So you're going to have a bunch of these guys all together. And as I say, you need 20 or 25 of them together to be your transmembrane domain. And so if you sort of have a program that scans along and looks at sort of the hydrophobicity locally, which is called hydropathy, it will go high at a certain point, and in the transmembrane domain, it'll stay high for about 20 or 25 amino acids. So if you look here, this is residue number. This is, these are in increments of 10, right? 50, 60, 70, 80, 90, 100. So what you see here is this starts at about 70 and goes a little past 90, so that's a little more than 20 amino acids, so that's the right one. So this basically went hydrophobic and stayed hydrophobic for long enough, so it's probably a transmembrane domain. And in fact it is, it's from glycophorin. Now, sometimes when you look at a protein and you do that same thing, you'll see multiple ones, and that just means that this protein has multiple transmembrane domains. So this is, for instance, rhodopsin, and it forms this sort of structure. And it goes back and forth. Now that's important because if you think about an alpha helix, there isn't really much space inside of an alpha helix for things to come through. So if you're talking about creating some sort of internal space in the protein to have stuff go through it, you're going to need typically multiple alpha helices which can sort of form some structure together, and then they can have an internal region which is different from the external region. So typically things that are going to act as transporters that allow things to go across the membrane will either be multiple alpha helices or they're going to be beta barrels. And I think there's a picture of a beta barrel structure in your book. So that sort of is what we have to say about these guys. But before we go on to the nature of all of these transporters and how they work, I want to talk about another set of things, which is glycosylation or, well, glycosylation. So when you make proteins, they're just amino acids, right? You make a sequence of, you take the DNA, you make your messenger RNA, then your ribosome comes along, makes your amino acid sequence, and so then the amino acids fold and you get some polypeptide natural protein. And those have many great and useful properties, but they don't have all the properties that you might like. So in particular, proteins are relatively sensitive to things like proteolytic degradation. So you can have proteases that come in and chop them up. Okay? And so there are many cases where you don't want them to be sensitive to things like proteases. You want them to be resistant. In particular, if they're on the outside of the cell, and they're sort of protecting the outside of the cell, they're helping to form the barrier of the outside versus the inside, you might want to give them a little bit of extra help and a little bit of armor. And the way you do that is you add carbohydrates and sugars and lipids to these guys, and that process, that process is called glycolysis, and then the proteins, once they've had these carbohydrates and sugars, then they're called glycoproteins. And if you add lots and lots, and so there are in fact more of these carbohydrates and other things than there are protein, then they're called proteoglycans. So proteoglycans and glycoproteins are the same thing, but these guys have more protein and these have more glycan. Okay? So these proteoglycans and glycoproteins can have all sorts of useful functions. In particular, they can really change the sensitivity of these proteins to being degraded or altered, and they can also affect the ability of things to bind to the surface of the cell and get inside. So and we'll come back to that in a second. And all of these modifications of these proteins happen in the Golgi. So when you make proteins, we'll be talking about this later in the course, but when you make proteins, you start off making them maybe in the cytoplasm or in the ER, but the ones that are going to be sent to the outside of the cell, they all end up in the ER, and they start getting modified in the ER, and then they're transferred over to the Golgi, and they're modified even more in the Golgi. And it's in the Golgi where you have all of these proteoglycans made, where the glycoproteins all the carbohydrates are added to the proteins, and then these guys are stuck into the plasma membrane from the Golgi. So it's in the Golgi that all of these things happen. Okay, so what does glycosylation do? Well, I told you it can protect the cell from chemical and mechanical damage. It can keep other cells at a distance and can control cell-cell interaction. And it can prevent undesirable cell-cell interaction. So for instance, if a bacterial cell wants to get inside, various forms of glycosylation can help make it hard for the bacterial cell to bind or for the bacterial cell to get inside. And it can also contribute to things like blood clotting and inflammatory responses. So we're going to see, in particular, we're going to be talking about blood and blood clotting and such in our next little bit. So that brings us to blood. So you've got all these red blood cells, and they're floating around in your body carrying oxygen. But they're also the target of many infections. And so there's this system of antigens that has been recognized for a long time which relate to this, and we're going to talk about. So the fact that not all blood is the same was discovered in, I think, the 1920s. Maybe it was the 30s. And what was discovered basically, so originally they would do transplants, like on battlefields and such, where someone would be wounded and they would need some blood. And the problem was that frequently, when you got a transplant, you would die after it because you'd have a really severe immune reaction against the blood, and that would pretty much be the end. And so they figured out in the 20s or 30s that what caused these very severe reactions were that there were actually 3 different types of blood, or 4 different types of blood. There was some blood that basically didn't have any antigens on its surface, and there was some blood that had A antigens and some blood that had B antigens and some blood that had both. So what does that mean? It means basically that if you took the blood and you mixed in what was called an A antibody, the A antibody, if it was O-type blood, which didn't have either one, the antibodies wouldn't bind the blood. But if it was A-type blood, if you mixed it in with the antibodies, the A antibodies would bind, but the B antibodies wouldn't. If it was B-type blood, conversely, the B guys would bind, and if it was AB, they would both bind. Now what happens when the antibodies bind is that they cause it to clump up, and so you can tell whether they bound by how much clumping there is. And so, let me see, I can show you a picture. So this is normal blood, and when you put in anti-A antibodies and it's A-type blood, you see that. So it's pretty clear under the microscope that it's been clumped due to the A antigen. Okay, so that's all fine and good, but how does this relate to your immune reaction? Well, one of the things that your immune system does is it's constantly trying to figure out self versus other and leave self alone and attack other, right? I mean, that's what your immune system is supposed to do. So if you have O-type blood, that means that your blood cells don't have either A antigen or B antigen on the surface of the blood, right? So you're happy when you see O-type blood, whether it's from yourself or from someone else, but if you get a transfusion of A-type blood, then your immune system suddenly recognizes that there's an antigen on the blood that isn't self. Okay, and it's very unhappy about that, and so it mounts an immune response against this massive infection from the A antigen. Okay, and so now you just are attacking all the blood that you've got. So that means that if you're O-type blood, you can't get a transfusion from A-type blood because you're going to recognize it as other, and you're going to mount an immune response. But now let's suppose you're A-type blood, and you got O-type blood. Well, A-type blood has the A antigen. When you get the O-type blood, it has neither antigen, so your immune system has nothing to recognize because this doesn't trigger anything, so you're happy getting the O-type blood. Okay, so what all this means is that O-type blood, because it doesn't have any antigens, is a universal donor. It isn't recognized as a problem by any of these guys. If you have AB blood, then you recognize both A and B as normal, and so if you get blood with A or B, there's no problem, and so AB is the universal recipient. Okay, so that's sort of, from a simple level, why, what the difference is between these blood groups. But now the question is, what's really going on at a molecular level? So it turns out that on the blood cells, there's a single protein that is a glycoprotein. And it starts off basically being modified so that it has a number of sugars and such, but it ends in this galactose with this fucose here. So this, and basically if nothing else happens to it, you have O-type blood. Okay? But there's this protein that's called A1 glycosyltransferase. Now, A1 glycosyltransferase can have 3 different forms. Okay? A1 glycosyltransferase can either be you can either not have it, or you can have a dead enzyme. So the A1 glycosyltransferase transfers a glycosyl group onto the end. So if you don't have this as an active protein, basically all of your blood stays this way. But if you have it as an active enzyme, it can have two alleles. One allele tends to transfer this galactose. And when you have this galactose transferred, you have B-type blood. And the other allele tends to transfer this GalNAc, which is N-acetylgalactosamine. And so if you have this allele, then you have A-type blood. Okay? So a single mutation, well, a mutation in a single gene is responsible for the three different blood types. And AB blood is basically when your mother was, say, A-type and your father was B-type, and so you get both versions of this gene, one copy from each. Yeah.
So the glycolysis transfers these sugars, these residues from the O antigen to make either B or A.
Yeah, they add the residue onto this guy. I'm sorry, yeah. So they basically, you go from here to here, you go from here to here, by this glycosyl transfer is adding the galactose or the GalNAc onto the end, yeah.
Based on the mutation in the gene.
Based on, yeah,
Okay.
based on your allele, that's correct.
But it starts at the O.
You start it with O and then,
Okay.
and then you modify or not.
Thank you.
Yeah. Okay, so now that's sort of interesting, but the question is, so why? Why do you, from a sort of historical evolutionary perspective, but also, so what does it do for you to have A-type blood or B-type blood or O-type blood? Okay. So it's a combination of selection and founder effects. So how many people know what founder effects are? Okay, so 2 people. So what a founder effect is basically is if you've got a population and they start from a few founders, then the founder's genetic information is going to control the entire population, right? So if the founder had a particular type of blood, all of the offspring are going to have a particular type of blood in general. And so one thing that is going to affect the particular blood type of a group in a particular region is where they came from, sort of who the founding members are. But then in addition to that, you can have selection effects. So we are under selection, and in particular, if there are particular diseases that are particularly bad if you have a particular type of blood, you're going to get selection, which makes you much more likely to survive if you have one type of blood or another. And so it turns out that both things are going on. And there's some very interesting differences between the blood types. So basically, A and B and AB tend to clot better. So that means that you tend to bleed less from a wound before you clot up if you have A or B. What's more, it tends to mean that with sort of no medical care, women who are A or B are probably going to survive childbirth better because they tend to have less complications due to pregnancies as far as bleeding. Okay? So AB is great. So then why isn't everyone AB? So what I've got to stress here is that about 63% of the world is O, 21% is A, and 60% is B. Okay, so if AB is so great, why is so many O? Well, it turns out that there are a couple of huge selective pressures that have really favored O relative to AB. And one of those is that O makes you more resistant or able to survive malaria better. Okay, so why is that? Well, it turns out that if you've got malaria, one of the things that happens is in the cells that are infected with the malaria parasite, they tend to clump together and form these things called rosettes. And those clumps of blood can then get stuck and cause all sorts of complications and can cause you to die. Well, it turns out that people who have A-type blood tend to get these rosettes much worse, and they tend to be bigger and more sticky, and so they tend to have many more complications from malaria, whereas people with O-type blood don't get as sick when they're infected with malaria because the rosettes aren't nearly as bad. So people with O-type blood tend to be more resistant to malaria as far as surviving, and in places like Africa, where there's a high malaria burden, the majority of the population is O-type. Now, in contrast, you can, all of these glycosylations on the surface can give you protections against some things, and the additional glycosylation that comes to A or B can give you some protection against Yersinia pestis, which is bubonic plague, which was very prevalent in Europe. And so you tend to have, so because group O appears to be more susceptible, it seems that one of the reasons that you have more of A and B in Europe is that malaria was less of a selective pressure and maybe plague was more of a selective pressure. There's also, but A is somewhat more susceptible to smallpox, so in places where smallpox was important, then it looks like there's more B. So anyway, there's all of these trade-offs, and there are also various cancer risks that are different in different ones. So it's, I think, a very interesting area to try and understand how all of these things at a cell biological level affect the organism. There's a very nice review paper that looks at these things. It was published in 2010, and I put it on the website, so you can download it if you have any interest in reading it. I'm not going to test on it any more than what I've said here, but I think it's interesting. Okay. In addition to the ABO blood system, there's the RH system, so they're both relevant. They're looking at different proteins, and we're not going to go into it except that typically you'll see something like A positive or A negative or B positive or B negative, and that positive or negative typically refers to whether you have or don't have the D antigen in the RH system. So it's a separate set of proteins, and there is incompatibility between RH positive and RH negative. So if you have the D antigen, then presumably you can get Rh negative blood, but if you don't have the D antigen, you're going to mount an immune response if you get the D positive blood. And you can get incompatibility between mother and fetus, and if you want to sort of talk about the genetics that lead to that, I'll discuss it with you after class. Okay, any questions on that before we talk about transporters? Okay, so we're now going to talk about things going through lipids, bilayers. And we can have simple diffusion of small molecules, but larger things don't go through, so they need to be transported, and they can either go through a channel, or they can go through transporters, which basically bind them and undergo a conformational change and let them come out the other side. And so there are diffusive transporters, which just allow things to go down to concentration gradient, and there are active transporters called pumps, whose conformational changes are driven by energy input. And we're going to start by talking about transporters. So, transporters. Yeah, okay. So the simplest transporter is what we call a unitransporter, and that's going to transport things down to concentration gradient. A symporter or an antiporter couple transport of two different things, where one thing is transported down its concentration gradient, and the other thing is simultaneously transported up its concentration gradient. And so they're using the energy in the concentration gradient to drive the transport of the other thing. And antiporters are the same, but they're transporting things opposite. And then pumps basically use energy to transport things, and so they can transport things against the concentration gradient. Channels just allow things to go through, so they only work in favor of a gradient, but they can transport things much faster. So all of these are going to have multiple membrane spanning segments, typically alpha helices, to create that internal space that's different from the lipids. And so they're going to form this protein line pathway, and all of them are going to go some sort of conformational changes. The differences are the transporters, the uniporter is just diffusion, it's passive. The co-transporters are active in the sense that they're burning energy in the gradient to transport something against its gradient. The pumps are hydrolyzing ATP to move something against the gradient, but the channels are just passive and allow things to move down their gradients. So now let's talk about transporters. So the idea of a transporter is that it can be in two conformations, and it can bind stuff, and when it binds, say, the thing that's going to transport, which is bound specifically, then when it's bound both of them, it undergoes a conformational change, and when it undergoes a conformational change, now it can release them to the other side, but as soon as it releases it, then its favored state is back like this, so it releases them and then it flips back. It can sort of undergo both directions, however, so if there is not a concentration gradient, it's not going to lead to things on average moving one direction or the other, so it's driven, the rate of transferring things across is driven by the amount of the concentration. And the concentration, the transport rate is typically slow, which is about 100 to 10,000 molecules per second. Okay, what controls the speed? of the transport, well, there's sort of a maximum speed where no matter how much of the solute you've got, there's some intrinsic dynamics of opening and closing. And so the fastest this can go is if you immediately fill it up as soon as it's open, it's still going to take some time to go through its cycle, and that's going to set an upward time scale. So these guys are soluble, and no matter how much of this thing you have, there's some maximum speed that they're going to transport stuff based on the intrinsic cycle. But if you're below that rate, the concentration and also electrical forces are going to help speed you up. So the higher the concentration, the more you're going to go through. And if you're neutral, if there's no charge, if there's no charge across the membrane, it doesn't matter. But if there is a charge across the membrane, if you've got positive things that are being transported and inside is more negative, then these are going to feel a force that's pulling them in, and so it's going to be sped up. And conversely, if the electrical gradient is opposite the chemical gradient, it's going to slow you down. So if these are positive and they're going into a positive environment, then they're going to feel less force pulling them in, and you're going to slow down. So one example of something that's being transported is GLUT1. So GLUT1 is a transmembrane, a lot of transmembrane domains, and it basically transports glucose down glucose's concentration gradient. It's a unique order, and it basically works to transport glucose from the blood into the brain. Okay, so it's passive. It's facilitated diffusion, so it's down its concentration gradient, but it's faster than passive diffusion, and it's specific and it's saturated. So there's its saturation, and it's faster than that. So why is it? How does it happen? Why is it happen? So you have blood, and you have glucose that is in the blood. Of course, your nerve cells, your brain uses a lot of energy. And glucose is one of the things that mitochondria run on for energy. They, convert it to ATP. And so you need glucose to get into the brain for your mitochondria to produce ATP to run. And if you don't get enough glucose in the brain, then the neurons start dying or very unhappy, you have seizures. Yeah. So in fact, if you have a mutation in GLUT1, then you have a disease, and you can actually treat the disease by having a high-fat, low-carb diet. So why is it that a high-fat, low-carb diet helps in this? Right. So what do the ketone bodies do? So what are ketone bodies versus glucose? Right. Ketone bodies are lipids. So the key point is that your mitochondria can run on two different fuels. Your mitochondria can run on glucose, or they can run on fatty acids and lipids. Mitochondria can produce ATP from either one. But if you have a lot of carbs, then you tend to produce a lot of sugars, and so that favors the mitochondria running off of sugars. But if the sugars can't get across this barrier, then they don't get to the brain and it doesn't do any good. If you have a high-fat, low-carb diet, you're producing a lot of fatty acids, which go through the ketone bodies. But the key thing is that these fatty acids get into the cell through an alternate pathway, and the fatty acids can be used by the mitochondria, just like the glucose, to produce ATP. But the mitochondria are fine, so if they get the feedstock of the lipids, they can run off of that. So what you're doing here is you're forcing the mitochondria to use an alternate pathway to get their feedstock to produce ATP. Okay, we'll stop there and pick up next time.